know what's insulin, right? And but you may not have heard of insulin receptor. So, and today we're gonna discuss the relationship between these two, and also discuss what insulin will bring to the effect of insulin on cell in terms of metabolism. Yeah. So here's insulin signaling in a nutshell. Yeah. Very simplified version, and if you too small? No. Okay. So this is insulin receptor. It's mostly homodimer. Okay, and it can bind to 4 insulin. So yeah, you can also bind only 2, but the maximum volume of insulin is 4. Yeah, 4. Yeah. And it's a receptor tyrosine kinase. So if you guys know other kinds of receptor tyrosine kinase, like EGFR, right? EGFR dimer can only bind to 2 ligands. So yeah, you can see that it's quite different, although they are same kinds of receptor. Yeah. And for insulin receptor here, this is called type 2 RTK receptor tyrosine kinase. So moving on, let me show the pointer here. Okay, so you can see that in the extracellular domain and intracellular domain, and where the phosphorylate happens in the intracellular domain, I believe Campbell has this, right?
Yeah.
Okay, and then oh. Okay, then for RTK signaling, we will have to be facilitated with a adapter. Here it calls insulin receptor substrate. Here, IRS. Okay. IRS recognize these phosphorylated tyrosine, and then can be phosphorylate and recruit downstream effectors. Yeah, I believe that's what it calls. Yeah, like effectors.
对呀，要拿 IRS 去装那个 phosphorus。那 IRS 它就是一个 adapter， 就是它会接在那个 RTK 的。我知道，但是对，那个 IRS 跟 PI3K 中间。那个就是一样，也是有一 P 而已。因为 IRS 被接之后会被零商化。对啊 ，IRS 被零商化之后，它会有很多个零商化位点，就是我记得它还有不同 type 不一样，然后我记得好像有些可以到3个。还是怎样的，反正它是一个 adapter， 可以把它想象它是一个接零商化。然后 PI3K 的 PI3K 是那个把 PIP3， 就是我意思是它去辨识这个零商化位点。
Yeah, it should say that activated IRS can then activate PIP.
就是基本上是它被零商化之后，它是把零分丢给它。那上面没有写它怎么。它是那个 IRS 去接它上面的零分，然后。
So PI3K is a substrate of IRS.
Yeah. And 中间那个磷酸基是谁做的
this one belongs to, I think.
应该是 IRS 被零商化，都再把零商化丢给 PI3。就是它可能被它可能接到零商化之后，它自己活化，然后它自己活化之后可以让它自己被零商化，然后它被零商化之后再把零商化丢给PI3。而且 IRS 有自己零商化的功能。不是自己零商化，我不知道是自己零商化或是别人零商化的，但是直接自己的。
Actually, I think.
那我现在查。
I think. Only here PI3K is phosphorylated.
因为这最中间啊，我不知道它到底是在画。
Exactly. But either way, you get the important point that IRS can phosphorylate PI3K, right? Yeah, and I think that's a key point here. Yeah. So, and then this PI3K can phosphorylate phosphoinosine diphosphate. Yeah, it's a inosine. Yeah, you might know that.
Just PIB2.
Yeah. Exactly. So this phosphorylated activated phosphoinosine triphosphate then can activate. Here it should. Sometimes you see the name AKT or PKB. Yes. Okay, so they're kind of a PD kinase. I forgot what's the what's the name of it? Yes. And they all belong to a family of kinase called PDK, but phosphoinosine dependent kinase. Okay. So let's call this AKT because it's more common.
this是 AKT，
This one is AKT, yes.
我看它下游，对，这是 AKT 的下游。
Yeah. So then AKT here activate mTOR and it facilitates expression of GOT4. I mean by here, expression means to get it on the cell membrane. Yeah.
对，是一个 special， 然后它会丢上去嘛，这个是花这个事情。所以它的下游，所以它传下去的东西，它在一个恒定温度下被就这个环境，它就是可能会促进它这个楼层。
So you put a vesicle on the membrane, yeah, because GOT4 here is a glucose transporter. Yeah.
这样它是活化成什么器啊？那个
真的那我厉害。
Maybe like hedgehog signal. It also has a step where you have to move a vesicle up to membrane. So yeah, in here they mainly activate some protein that interact with cytoskeleton, such as kind of forget, but mainly these activated protein can interact with cytoskeleton and make the thymine move to and fuse the membrane. Yeah. So that's part of that's the GOT4 part. So you know that when insulin kick in and this glucose transporter will be expressed onto the cell membrane. So exogenous glucose can flow into cytoplasm. Okay. And then this is glycogen synthase kinase. Glycogen 肝糖。
IRS 是被那个 insulin receptor
酸化。Phosphorylate. Okay. Good.
对，它的在 C 端。
你在讲什么？
它就是那个我想想，就是 insulin receptor 它会形成化 IRS 的 C， 然后
越来越少
的那一个我自己猜，反正就是它是被 insulin receptor 给影响化。然后可以看BI3K 它是接到。对啊，他可能就是他被影响化，都会把你的 schedule 对。
This meant, glycogen mainly synthesized in liver and
in the start site.
like a primer, right?
还有一个他要先成盒。
Yes. And so, generally it's some that's way ahead of what glycogen synthesis kinase can control. Yeah. But I think that is pretty interesting part about glycogen synthesis, and mainly glycogen synthase can, I think, only elongates the glucose chain, and you need another enzyme to branch it. Yeah, so glycogen synthase can do the branching, I think. Yeah. And when this kinase is phosphorylated, this effect is inhibitory. Yeah. So you can imagine that without insulin, this unphosphorylated kinase is activated, and it can phosphorylate the glycogen synthase. Yeah, so make the enzyme lost its activity. Yes. Okay. And then there is mTOR. Yeah, it's also a downstream target of AKT. Yeah, and mTOR is also a very important hinge in the overall control of metabolism. So yeah, maybe next time we will talk about mTOR. Yeah, and so mTOR here mostly involved in lipid and amino acid protein metabolism. Yeah, and when the insulin kick in, the effect will be the increase of protein synthesis and increase of lipogenesis, which means to generate lipids. Yeah, and then decrease of lipid catabolism. Yeah, decrease of lipid catabolism.
这是糖精性症吗？我猜一下，还是不是？就是单纯的脂肪那边，它会把脂肪的那个减慢，然后它可以促进脂肪的。哦，你说 decrease，它是 decrease 合成
decrease
分解
catabolism，
然后合成那个。
同胞遗忘。Okay, then here's a foxo. Foxo is a family of transcription factors, and they mainly deal with stress and generally some other aspects of cell cycle. And we will also discuss about it in next time. And so here you can see that what insulin do is to make glucose in our circulatory system into cell, and then increase the rates of anabolism, decrease the rate of in like storage organ like liver, it will decrease the rates of catabolism. Okay. And then it also serve as a survival signal because here the foxo mainly deal with stress response. So and ultimately it will lead to something like apoptosis or other way of your cell just die. So here by inhibit foxo, insulin can also serve as a survival signal. Yeah. Then there is this half of signaling pathway, which is Ross, Schwab, Meek, Erk, and this also is sometimes you will see as MAPK, right? MAPKK, sorry. Yeah. Then downstream will be MAPK.
就是它是一个 MAP， 就是有丝分裂源激酶，然后它是一个，就是它应该说它这个路径是跟生长调控有关，然后它基本上会有 MAP KKK MAPK MAPKK 跟 MAPKK 就是
Yeah, kind of.
上有个 KKK， 反正就是它有这
，this
三个
one's KKK。
就是那个 KKK 是 kinase，
就最下面叫做 MAPK， 然后 MAPK 的基酶就是 MAPK， 然后对，我们就叫它 MAPK 开始。
Mainly what they do is something like cell cycle passage, yeah, or other stuff like that, yeah, that can promote your cell's growth and to
然后
mitosis.
它下游就是一些 NH 卡巴比之类的东西。
Yeah, there's more than that, but mainly yes. All right, so what we have here is this one, this mainly about metabolism. We call it PI3K, PI3K AKT axis. Yeah. So this part of metabolism, this part of signaling, and this part we call it MAPK axis. Yeah. They do different things. Okay.
我好像找到那个它怎么把它调整。简单说就是，大概知道 snare 是什么东西，反正就是
PPT 大师哎，
大概就是AKT 被活化之后呢，它会去抑制 ASE60，那 ASE60就是我先讲 RAB 是什么 ，RAB 它就是一个，你可以把它想象成它是一个在运输囊泡上面的标记，就是有点像是门牌。对，就是不同的 RAB 会跟不同的 receptor 接，然后就需要不一样的地方。那接下来讲一下 snare， 那 snare 就是我的囊泡要送到膜上，那我然后送到膜上进行膜融合的时候，我会用就是一种用阿法螺旋组合起来的蛋白质，然后借由旋转拉开，让膜质做融合。对我讲的这很简单的部分，然后我们 snare 可以拆成两个部分来看，一个是 T snare， 一个是 B snare。那 T snare 叫 target snare， 就是说我们现在是长在膜上，那它里面有 transfer snare。哦，它是 target snare， 就是 target， 或是 transfer 都可以啊，但是大家都叫 T snare。然后 B snare 就是 physical snare， 就是长在囊泡上面。然后反正就是然后它们中间会有一个东西叫做 MUNC18。那 MUNC18这个东西它是我们的 T snare 其实有两个，一个是Snake25，它跟
什么新的？
对，然后就是 MUNC18是协助这个东西制作，应该说它就是如果你就想，如果它待在上面的话，它就会去保护那个它的 off 的一个构型，去防止这两个东西撞在一起。那它的离开会需要 RAB 的协助，对，反正就是，然后我刚刚说 AKT 会去抑制 ASE60嘛，那 ASE60它负责就是把 RAB 从 GDP 变成 RAB 的 GDP。对，反正它是抑制这个回去的阶段，所以它就可以让 RAB 去跟它结合，然后打开 snare。
它是让 RAB 接 GDP 还是？
它是 ASE60是把 RAB 的 GDP 弄掉，就是它是对。
Jack, gap, sorry.
靠没有。
Jack 是把 GDP 换成 GDP 换成 GDP。
Yeah, so it should be gap, right?
对，对，反正就是这样。反正就是它是负责它的膜。
今晚你知道有一点身高有一点，他直接考那个。
好几都有。好几都有 snare 啊。
他有一题直接穷纯英文，用名字。
有一件2016初赛之类的吧。Okay, so thank you for the detail. And move on.
Yeah.
Okay. So in here is basically insulin can the mammalian cell all have insulin receptor, but mostly it appears in liver, adipose, muscles, and yeah, sometimes tumors. Yes.
天文画出来，它不是一个正常的东西。
And you can see there is different effects of insulin here. So, but mostly it is about nutrient storage or nutrient intake and survival. So that's why tumor showed up. Yeah, and
你要想，如果 cancer 它可以自己做，它可以自己去 activate 这个 RTK， 或者它自己可以弄一个很神奇的 RTK 到它的模式，它就可以自己去接这些讯号，然后自己产，就是甚至它是自己去把 RTK 下的路径自己活化。
他只是想要拿那个 glucose，
哈？
他是想要把 glucose 抓。
还有，但是 RTK 跟 survival 有关的。
survival is also important. Yeah, yes. Exactly. And in brain has constitutively expressed glucose transporter. Yeah, so we don't need to express GLUT4 additionally. And what insulin does is mainly about like a neuron it's sort of like a neurotransmitter. Yeah. In the hypothalamus neurons, it can control your appetite. Yeah.
Really?
Yeah. Indeed. Yes. Mm-hmm. So.
什么时候的研究？
还不知道，对，下次就。It's a long time. It's a long time ago, and they do it by injected insulin, or maybe by spraying it in your nasal, and just to isolate that from your body and so on. Yeah, to make your
Jesus Christ.
Yeah, that's really cool technique. And they find out that when you apply additional insulin, it will make people eat less, and mostly men. But for women, they have, this is kind of not the case for women. And recently, there's a research about the insulin signaling in brain, and the relationship between menstrual cycle, 月经周期 yeah. And it's a pretty cool research. I suggest you guys to read it if you're interested in how insulin controls the appetite and controls our kind of food intake and other metabolic aspect. Yeah.
在脑里面的这个 receptor 那个会长不一样吗？感觉需要。
Not really.
Well,
what do you guys think?
我觉得它 pathway 至少要长不一样。
但是我觉得它的 pathway 其实还好，因为其实你 RTK 下游你接完不同的 IR 是可以接不一样的，啊，所以对吧
So
这它引发反应
而且其实我觉得 cell 上面 pathway 它并不是一个很单一的东西。
确实。
对，而且它可能会跟你这个细胞里面平常表现出来的蛋白质来去决定说我到底要走哪一条。所以我觉得你有可能都一样，反正你也知道 AKT 底下很大嘛，那你 AKT 被活化之后，你可能底下要被活化哪条路应该就该看。
可是我觉得 neuron 如果是 neuron 的话，它应该会变蛮多。
对啊。
Actually, we only find maybe one kind, one gene of insulin receptor in human genome. Yes. But why it acts differently? Mainly because you have different kinds of insulin insulin receptor substrates, and here, as Warren just said, that take FOXO here as example. It can it's a transcription factor, but in brain and in other tissue, it can express different kinds of gene. That's pretty reasonable, right? Yeah, because you have
因为 transcription factor 它并不会只说我只单独去活化。
in terms of the chromatin structure, you have heterochromatin, you have euchromatin, and